On behalf of the UK Lymphoma Clinical Studies Group of the NCRI and of the Swiss SAK Group, I'm very pleased that we're presenting some of the first look at the REMoDL-B study. And this is a big phase 3 study for patients with diffuse large B-cell lymphoma who've previously not been treated. So we recognise that actually diffuse large B-cell lymphoma is probably not just one disease, but very many different diseases. And there are distinct molecular groups, molecular phenotypes of diffuse large B-cell lymphoma. One of those is called the GCB group, and one of those is called the ABCs. And they have different oncogenic aberrations that result in the phenotype. And what's really significant about this is that they have a different clinical outcome. And the ABC subgroup has retrospectively done much less well than the GCB subgroup. And one of the things about the ABC subgroup is that the different genomic aberrations result in constitutive activation of the NF-kappa B pathway. So we hypothesised that if we could switch off the NF-kappa B pathway, in addition to giving these patients conventional R-CHOP chemotherapy, we might be able to improve the outcome of the ABC subtype. So we used a drug called Bortezomib, which has been widely used in hematological oncology. We added that to R-CHOP chemotherapy to test this hypothesis. So that was the kind of the plan of the study, but one of the key things about this was how do you define these molecular subgroups, ABC or GCB subgroups? And conventionally, immunohistochemical techniques have been used. However, they have been proven to be not necessarily reproducible. So we endeavoured in this study to set up a whole network so that we could do real-time gene expression profiling, developing whole transcriptome analysis, and particularly information about certain genes that define whether a patient's tumour was of an ABC or a GCB or an unclassifiable subgroup. And what we did is this was using formalin fixed, paraffin embedded, just the usual blocks that normally go through the pathology lab. And we devised a classifier that we could assess this in real time. So we recruited 1,132 patients, of which 1,085 of them we were able to perform gene expression profiling. What was amazing was that the success rate was really high, and only in 12% of cases were we unable to get a gene expression profile to define the cell of origin in these patients. So very high. The turnaround time was very quick, and we were able to get gene expression profiling from both core biopsies and also from surgical resection specimens. So obviously performing a gene expression profiler takes some time. The median time from patient registration was about 10 days. So actually all patients, when they were given one cycle of R-CHOP chemotherapy whilst the gene expression profiling was being performed, and by the time they were due their second cycle of chemotherapy, they were randomized, depending upon cell of origin in stratification, to continue R-CHOP chemotherapy or R-CHOP with the addition of Bortezomib. So we present a really early look at the data, and the data by the molecular subclasses of patients. And we can see that the demographics of the ABC population are slightly different from the GCB population. For example, they tend to be significantly older than the GCB group. We've also looked at the response rates to R-CHOP and R-CHOP plus Bortezomib, not broken down by arm, but the whole study group. And we could see that in the different molecular subgroups, actually there wasn't a difference in overall response rates, or even at this first look. We can see that the whole study population has an excellent progression-free survival and an excellent overall survival. And then when we broke that down according to molecular phenotype, although the follow-up is only still a median of 16 months, so quite short, we can actually see that the curves are pretty much superimposable depending upon cell of origin. So I think our kind of conclusions, we haven't presented by study arm yet, we need to have more events, but our conclusions is really that real-time gene expression profiling is absolutely feasible. You can do this in right now for patient benefit. We saw that we probably weren't preventing early failures by the addition of Bortezomib to R-CHOP chemotherapy, and I think we were surprised to see how well the ABC subgroup did when we anticipated they were going to be doing less well. And that may be a function either because of it's a prospective evaluation rather than a retrospective, as I'd done previously, or Bortezomib may be having some effect. But really, very exciting. It didn't appear that the addition of Bortezomib significantly added to the toxicity, and we await next year the presentation of the per arm analysis. I think the major challenges were, first of all, establishing a classifier to determine whether patients were ABC or GCB, and we had quite a lot of work to do that. But one of the big challenges was, at the beginning, was trying to get sites to pass on their formalin-fixed paraffin-embedded blocks to the central laboratory for analysis in a very timely fashion. And initially that was a bit slow. We spent a lot of time engaging pathologists and engaging the pathology teams so that this could happen. And by the first few months after initiating the therapy, absolutely everybody was, the study, everyone was on board. So it was challenging to set that up in the first place. But actually, once we had got that all established, actually the whole pathway flew very, very, very well, really. The next steps, we'll wait for a per arm analysis to see what the impact with Bortezomib is. But actually what we've done in the UK and with the SAK group is we've established a really important platform to show that with other novel agents we can add to R-CHOP chemotherapy, and we can stratify based upon expression profiles an individual patient's diffuse large B-cell lymphoma. So it's given us a superb diagnostic platform for future evaluations of other drugs.